All right, today we're going to talk about some of the mechanisms and details of what it takes from when your brain sends your muscles the signal to move and how that all goes on. So we're going to go through the mechanisms of what's happening at the cell membrane, the action potential, to the alpha motor neuron, and then to the skeletal muscle, the cross-bridge cycling, muscle contraction, relaxation, and so on. So first, let's start with the resting membrane potential. In order to understand action potentials, we have to understand the resting membrane potential and how that works and why it even exists and how. So let's get into it. First, we'll just draw a circle, and we'll call this circle a cell. And let's say this is a normal cell, has a phospholipid bilayer right here. And this cell has this concentration gradient, right? As you remember, the inside of the cell, right, is negative and the outside of the cell is positive. Well, why is that? What's happening? What's creating this concentration or electrochemical concentration gradient? Well, we need to understand a few critical points about this. Number one, in order to get this gradient, this entire gradient is really due to the differences in concentrations of ions, specifically potassium ion and the sodium ion. So let's talk about why that is and how it happens. So first we have our cell, and inside of our cell we have a lot of negative anions, right? And some of these anions are proteins that are impermeable to the cell. So as remember, we have different proteins here. We can imagine we have like this protein here, and it's a carboxylate group, so like aspartate or glutamate, for example. But notice this is a negatively charged protein, and that's one reason why the inside of the cell is going to have this negative charge. This is not for the resting membrane potential, but we need these negative charges in order to develop the electrochemical gradient. Okay, but this is impermeable. So this protein is like a integral protein or a transmembrane protein, right? It's impermeable, so it's not going to go out of the cell, but it's giving this negative charge. Then we have other types of molecules, such as ATP. Okay, well, ATP actually is very negative. It's a 4- charge, right? And then you have ADP. I'll just write that because when you break down ATP into ADP, that's also negative. That's a 3- charge. And then AMP, that's a 2- charge. So a lot of your phosphates is going to give you a lot of negative charge. But this is, of course, is permeable, but a lot of negative ions in here. And you have other ones like bicarbonate or lactate, for example. So lots of negative charges inside of there. Now, this is going to be important to set up that concentration gradient of potassium. If we remember that potassium is positively charged, and these are all negatively charged, so they're going to be attracted to each other. So if we put a whole bunch of, so first, the cell in normal, it is electrically neutral, okay? Because you can't violate the principle of electroneutrality. So everything is neutral. But what you have is these very important pumps right here. Okay, this is a sodium-potassium ATPase pump. And what it's going to do, it's going to pump ions. It's going to pump two potassium ions inside the cell, and it'll pump out 3 sodium ions. Okay, so 3 sodium ions are going out, 2 potassiums are going in. Now, that's going to be important because already we can see a difference in charge distribution. Because as you can see, with 2 positives going in and 1 going out, that's a difference of a difference of 1, right? It's a net charge difference. So that's partly what's contributing to the resting membrane potential. So this pump is going to continue pumping in those potassium ions to balance all this negative charge. And that's going to happen once we get to a concentration that's pretty high. So the inside of the cell is going to have a potassium concentration, okay, of the inside of about 150 millimolar, okay? So pretty high concentration. Whereas on the outside, our potassium concentration on the outside, okay, so out, is going to be equal to around 5 millimolar, okay? Now sodium, right, we're putting sodium on the outside. The concentration of sodium on the outside is going to be around 145. So sodium ion on the outside is equal to 145 millimolar, or milliequivalents. And on the inside, what is our sodium? So sodium, again, on the inside, going to be around 15 millimolar, all right? So very big concentration gradients. Then we have other major ions such as chloride. I'll just write here. So chloride, which is negative. This one is going to be around, let's say, I don't know, 10 to 20 millimolar, all right? And then on the outside is quite high. So this would probably be around 120 millimolar, okay? Don't get confused with that 3 there. All right, so this is the concentration gradients that are occurred because of the sodium-potassium ATPase pump, right, which is requiring ATP. Now we have that concentration. Let's consider what's going to happen if we have a whole bunch of potassium on the inside of the cell and not very much potassium on the outside of the cell. Where is that potassium going to want to go? It's going to follow its concentration gradient and diffuse out of the cell. So it wants to go out of the cell. And fortunately, it actually can do that because we have what we call these potassium leak channels. So in fact, I'll use a different color here. And I'll do it like this, and we have these potassium leak channels. And so the potassium can go outside of the cell. And we actually have quite a bit of these channels. And so all this potassium is leaking out. And it's going to keep on doing that until it reaches an equilibrium. Because remember, the inside of the cell is negative, right? It has these negative charges. So it's not just going to keep on spilling out indefinitely until it reaches, so say 150, so like 75 millimolar here and 75 millimolar out there. It won't do that because there's a lot of negative charges here. So the negative charge is pulling it back into the cell, but the concentration gradient is pushing it outside of the cell. So eventually, it's going to reach some sort of an equilibrium, right? And what is that equilibrium? Well, I'll show you how we determine that. But I want to say the same thing's happening with sodium, right? The sodium on the outside is very high, 145 millimolar. It wants to come inside of the cell. Plus it's negative. Well, a little bit of it can because there are also these sodium channels. Okay, so I can just write right here, for example. Sodium channels are, we call leaky sodium channels, allowing some sodium to enter in. But it's not a lot. Okay, the cell is mainly permeable to the potassium. So a little bit of sodium is entering, but a lot more potassium is able to leave the cell. Now, the question was, what is that final concentration once it reaches this equilibrium? And that can be determined by the Nernst equation. Okay, this allows us to determine the actual voltage potential of the cell membranes. Because remember, we literally are creating a voltage. Remember, voltage is simply the potential difference. And here, the cell membrane, this is acting as the insulator, the resistance. And then we have a charge difference on both sides. And now that's going to give us our voltage potential. So the Nernst equation, if maybe you've heard this before, is this simple E equals E naught minus RT over ZF times the natural log of Q, okay, which Q is basically the equilibrium that we're talking about, right? So in this case, it would be the concentration of ions on the outside over the inside. Now, the E naught is the chemical equilibrium of when everything is balanced. Well, in this case, it's going to be 0. Now, the R is the gas constant. T, we can use the body temperature, 37 degrees Celsius. And Z is the charge of the ion, and F is Faraday's constant. When we put all these numbers in, this equation can be reduced to the following. E equals about 61 for body temperature over Z, and then we can convert the natural log into base log 10 of log, and then as I said, this could be the concentration of the ions. We'll do the outside over the ion concentration of the inside. Okay, now take the log of that. Now, if you have your calculator, you can go ahead and do this, and let's do this concentration let's do what is the Nernst equilibrium for potassium? Well, let's figure it out. So potassium, right, is going to be equal to 61 divided by Z, which the charge on potassium is 1, so it's just 61, and then you take the log, you take the log of, we put the concentrations in here, so we'll put 5 over 150, take the log of that, multiply it by 61, and what do we get? We get the chemical concentration gradient is equal to about negative 90 millivolts. And that's why you've seen so many times that rest membrane potential for potassium is negative 90 millivolts. Now, in neurons, as you know, that rest membrane potential is often closer to negative 70 millivolts. And there's a reason for that, because let's do the same thing for the sodium concentration gradient. Okay, so let's plug in those numbers for sodium, right? So you would put on the outside for sodium 145 divided by our 15, and then you take the log of that and then multiply that by 61, and you would get the sodium concentration gradient, okay? Sodium concentration gradient would be around positive 60 millivolts. And that's its equilibrium concentration gradient. So again, if you were to just let the cell follow its concentration gradient because like the potassium wants to leave, but not leave completely because there's all the negative charge that wants to come back in, it would do that until it reaches negative 90 millivolts. And the same thing with the sodium. If you allow the sodium to just rush in until it reaches its concentration gradient, you would reach around 60 millivolts, all right? Now, because of that, a neuron is going to have a not negative 90 millivolts, but it'll be closer to around negative 70 millivolts because, again, some of that sodium is entering in, causing a slight depolarization of the membrane potential, which results in around negative 70 millivolts for a neuron, versus skeletal muscle, as we see, is closer to negative 90 millivolts. Now, as you can see with the Nernst equation, we only used one ion at a time, but of course we have a lot of ions going on here, right? Chloride, even calcium. And there's another equation called the Goldman-Hodgkins-Katz equation. And this equation is just modified of the Nernst equation, but it considers all of the other ions and the permittivities, because as I said, potassium has a higher permittivity into the cell than sodium does. So there are constants you can use, and you know what the permittivity is specifically. You can put those in the equation, and when you do that for that specific cell, you get exactly, say, negative 70 millivolts. All right, so this is how the rest membrane potential is established. Now, again, the sodium-potassium ATPase is important because that's what's going to cause the concentration gradient that's going to be occurring. But it only contributes a few millivolts to the actual gradient itself. Even though you're seeing like one difference in charge, that's just one ion charge, right? And you'd have to have a full coulomb of ions in order to have an amp, and then if the resistance is 1, that's like basically 1 volt. So you literally have to have hundreds of millions of ions that are moving in order to just get a very small millivolt change. And that's why you do have millions and millions of this happening all the time, and that's why it contributes to around negative 3 to 5 millivolts. So the biggest part of the rest membrane potential comes from the potassium able to leave the cells and create that charge imbalance, right? Leaving the cells because there's so much potassium inside, so it's following its concentration gradient, but it's negative inside the cells, so potassium does want to come back in, so it's not going to leave completely. When it does so, it reaches that negative 90 millivolts, and the same principle applies to sodium, and the same thing even for chloride and all the other ions. So this is the rest membrane potential, which is crucial to set up the action potential for the neurons, all right? So now let's talk about the neuron, and again, we're going to walk you through what happens from the neuron all the way to the action potential in the muscle fibers, all right? So let's continue going over here. And we can draw our neuron, all right? So let me try to draw a little picture here. We can draw, let's see, kind of like this here. All right, this is not a very good picture, but I am not an artist, but I'm trying to draw the dendrite, basically, of the neuron. Okay, here's our neuron. We'll keep on drawing this out a little bit more. So this is like an interneuron, we'll say. Okay. All right, and then here's the axon, axon bulb here. Here the terminal. Here's the dendrites. Here we have like the soma body, right, with the nucleus and everything. Now we want to get a signal of this to propagate an action potential of this neuron and activate skeletal muscle signaling over here, which we'll draw next. So what does this look like? First, we've got to get an action potential that's going to be happening over here. And our brain is telling this is an alpha motor neuron, we're going to say, right, that's going to control the muscles. So we have neurons that are telling your body to move. So we can draw over here, we can put something like this, a little channel right here. And we want to cause a depolarization to get a propagation going on. And we'll have to draw another neuron. Okay, it's like another neuron right here. And it's releasing another neurotransmitter, neurochemical. So in this example, it's going to release, we'll say, glutamate. Okay, so glutamate is largely a stimulatory amino acid. And when it's released, it's going to bind onto this postsynaptic ion channel. It's a ligand-gated ion channel. And it's going to allow sodium influx to occur. Okay, so the sodium influx is going to occur. Okay, and that's going to cause this wave of depolarization, a positive wave. It's going to come down over here. And this is where we have a whole bunch of sodium channels. Okay, this is called the axon helix. Okay, this is basically the axon helix that's integrating all of the things that are happening over here with these dendrites in the soma body. Now, so glutamate binds to the ligand-gated ion channel. Sodium comes in, but it doesn't necessarily reach a threshold until it gets over here and causes the depolarization of these sodium channels over here. Okay, that's going to be very important to remember. But there's some competition that is going on, because on the one hand, where you have glutamate binding causing the depolarization wave, you have something else going on that's inhibiting that. So let's say you have another neuron or something over here. So you have this other neuron, and you have this channel over here. This neuron might release something like, say, GABA. Okay, so GABA or glycine, these are more inhibitory neurotransmitters. And when this binds to this ligand-gated ion channel, what can happen is you cause a chloride ion to come in, okay? So you have chloride influx that's occurring. Remember, chloride is negative. So what is that going to do to the cell membrane potential? It's going to lower it, right? It's going to bring it down. So chloride influx is occurring. That's going to basically start counteracting this effect. So this is inhibiting. This is exciting. So there's a term for this that I want you guys to know. We call this one an EPSP, excitatory postsynaptic potential. Whereas this one right here, we can call an IPSP, an inhibitory postsynaptic potential. And so again, so chloride is entering in. There's other amino acids or neurotransmitters could open up a ligand-gated potassium channel, and the potassium could come out of the cell, potassium efflux, again causing the hyperpolarizing of the cell, which counterbalances the sodium channel. So if you can imagine, it's time for you to study for your exam, and part of you is saying, I need to go study, I need to do that, and another part of you is saying, I don't really want to. So you have like two conflicting signals going on. I mean, it's kind of oversimplification, but it is kind of a tug-of-war between the EPSP signals and the IPSP signals and who's going to win out. And the goal is essentially to get the EPSPs to win out so you can get an action potential. Well, all of these waves, right, this is sending a negative signal, this is sending a positive signal, and then they're going to be the integrated or the summation is going to occur right here in order to induce an action potential or not. And in order to get an action potential going, over here, we need to hit a threshold of about negative 55 millivolts. So we can actually just draw a little graph here. So this is on the millivolts on the y-axis, and we'll just say this is time. And we want to hit, well, so we have our negative 70 millivolts, and we want to hit that negative 55 millivolts or so in order to hit the threshold. Okay, so this is our threshold one. Now you have the EPSP occurs, sodium's coming in, and it's going to, sodium rushes in, it's going to slightly depolarize the memory potential. Well, what's that going to do? You're at negative 70, so it's going to start bringing that charge up, right? But if it wasn't a very strong signal, then that charge is going to go back down, okay? However, you could do another signal, like boom, boom, like consecutively. And so when you hit that, then it can add this one, and then another one opens up, sending it more positive charges, okay? And this is going to sum, it's going to add up and get closer to that memory potential. But maybe at the same time, you have more of these hyperpolarizing signals that are coming. And so now this is going to bring it right back down, right? So there's this tug of war, like I say, going back and forth. And so this is the summation aspect. Now, if you have the same neuron that's sending the signal again, like faster and faster, higher frequency, okay, that's going to be called temporal summation. And so the exact same neuron, it can go boom, boom, fire, 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 and it's going to hit that threshold and then cause the action potential. So again, if it's the same neuron sending it, that's temporal summation. Another way you could do it is you could have a neuron. So you could have another neuron over here, and it's also releasing another stimulatory amino acid, or glutamate, I guess in this case, right? And it's causing the sodium influx to occur, and here's the positive charges, so you have multiple neurons that are doing this, and this will be called spatial summation, all right? So you have temporal summation and spatial summation. You want to understand those two different ways, and so, but together, if you get the temporal or spatial summation, or even all of them, together you can hit that threshold, and when you do, then boom, you get your action potential, right? And you get that depolarizing wave to come through, which is talking about right here what's happening with the sodium channel, and now we can get the action potential, okay? So let's continue finishing what's going to happen here. So you hit that threshold, right? And now you have all this sodium channel, sodium starts rushing through, okay? Now this is the axon helix where a lot of this stuff is being integrated and happens. This is chock full of all these sodium-potassium channels. Now over here in the neuron where the axon is, you actually have these little, I'll just put these here. Any guesses what these are? Trying to draw these, basically these myelin sheaths. Okay, so put some sodium channels. Oh, that wasn't very good. Some sodium channels right here. And notice these are all really tight together, but these ones are spread apart further. And you can do that because you have this myelin that wraps around that portion of the cell membrane and insulates it even more. So you're going to really increase the resistance. And as you increase the resistance, well, you could use like V equals IR, voltage equals the current times the resistance. And that equation or relationship allows you to see as you increase the resistance, you can increase the voltage. And now you can carry this charge down the axon faster and lose less voltage. So when you have a cell like this and you try to do an action potential, a lot of that voltage, those ions, they diffuse all throughout the stuff. You have like, mitochondria, the Golgi apparatus and all these other organelles and lysosomes and so on. So that charge is lost a lot over here. Whereas over here, guess what? This is like a really skinny part of the cell right here. In fact, you only have just this little actin type molecule over here for anterogate and retrograde transport of the little vesicles. You don't have like mitochondria, the nucleus, or all these other organelles in this place. It's just this axonic fluid, basically, and it's this really skinny part. So you can really carry that voltage very quickly, like hundreds of times faster than you can when it's not myelinated. And so this myelin is really important. And remember, it's the Schwann cells that produce this myelin around the cells. And that's in the peripheral nervous system. So they're still, Schwann cells are these modified glial cells. And of course, it's in the brain, the central nervous system, then they're just glial cells, or oligodendrocytes. So the Schwann cells are very important. You make the myelin sheath, and now the sodium, this positive charge, is kind of hopping. Just, this is basically, it's jumping, right? I was going to say saltar, like jumping, but we call that literally saltatory conduction. And it's salt too, so I guess that works as well. So you have this saltatory conduction from the sodium, and it just goes a lot faster because of the myelin sheath, and it's very fast. Okay, now this charge gets sensed over here, and now we have this other channel is very important, and this is a voltage-gated calcium channel. Okay, and it's going to allow calcium to enter into this into the synaptic bulb here, and that's going to be very important. But this voltage carries from the sodium channels will come here, activate this calcium channel, which gets activated at around 30 millivolts or so. I mean, just around that. So it's a positive, quite a positive charge. The calcium is going to enter, and here we have all these vesicles inside of this neuron, right? And these vesicles contain a whole bunch of these little dots. And these dots represent what? These dots represent ACH. And ACH is acetylcholine. And that's going to be released and bind to the nicotinic receptors in the neuromuscular junction. So this action potential is very important for this to happen. So we're going to talk about that here in just a little bit. But now I want you to understand, once we did this propagation, the action potential, now what do we need to do? We need to repolarize the cell. We need to get it back to where it was. And the way it does that is, as we talked about, with the potassium channel. So this cell also has potassium channels, which we can, well, that was right on top of the myelin, but that's okay. And the potassium is this, remember, there's a high concentration of potassium inside the cell, and so what's going to happen is, that's right, it's going to leave the cell. So you have the potassium is going to leave, and as it leaves the cell, it's going to quickly repolarize the cell. So you have the depolarization, the repolarization, and essentially it's even going to hyperpolarize the cell until eventually it goes back to resting membrane potential. Now, this helps us also to understand the terms of the absolute refractory period and the relative refractory period. So I want to talk about these channels a little bit more detail, what's really going on. And I think this will be helpful as we go through this. So let's write another. So this was our temporal and spatial summation. So our temporal summation of temporal, or we have our spatial. But that's still different than what we're talking about over here with these channels and our action potential. So let's go here, and I'll draw this graph here. We'll put our millivolt, we'll put our time, and again we have negative 70 millivolts. Well, actually, we want to show you something different here. We're going to go negative 90 millivolts, negative 70, because I don't need negative millivolts there. Negative 90, negative 55, and we can say 30. All right. And then again, this is our threshold, and this is our resting membrane potential here is at negative 70 millivolts. Okay, so when we get enough EPSPs in order to induce an action potential that it has to reach that negative 55 millivolts. So again, you have some your stimulus and EPSP going on here, and you start getting a positive charge. Oh, and then you get an IPSP, goes back down. Oh, but then you finally get another EPSP, and you get more of them. You get temporal and or spatial summation, and then you come in here, and then boom, you hit the action potential, and now it really comes up, shoots up very quickly, right? And then when you hit the repolarizing phase, so it'll come down, It'll go, now we're finally to negative 70 millivolts, but hey, it keeps on going even down to negative 90 millivolts, and then it starts coming back up, and then finally it's back to our resting membrane potential, okay? So what does that look like, and why is that happening? And it all has to do with these different ion channels. So first, once we hit negative 55 millivolts, that's the depolarizing threshold of the sodium channel. Now the sodium channel has, these voltage-gated sodium channels have two different gates, okay? So I'm going to draw a sodium channel right here. Let's see how I want to write this. Actually, let's see if we go like this, and it's kind of a fancy artistic way here. Here's our sodium channel, and this actually has two gates on it. So we have this gate right here, and this one is called the M gate. And then we have this one right here, and this one is called the H gate. Okay, so gate, and we have the M gate. And this is the inactivation gate and the activation gate. And at rest, at negative 70 millivolts, this is what this channel looks like. Now, once you hit negative 55 millivolts, that voltage is going to stimulate this M gate to start to open, right? So you have your sodium channel, and it's hit negative 55, and then boom, that M gate right here, it starts to open. So as soon as it opens, then all that sodium ions are going to rush into the cell. As it rushes into the cell, we cause very quick depolarization. But once we hit negative or positive 30 millivolts, this H gate closes. Okay, so that's another configuration of the H gate. So we have another way to look at this. So we have here, now we have the M gate is open, right, and the H gate is open. Okay, so H and M. And then we have another configuration where we have the, so now we have another configuration. So here where the M gate and the H gate were open, and then finally we're going to have the H gate is going to be closed, but the M gate is going to be open. And we'll talk about how this happens through this process. Okay, so again, you hit negative 55 millivolts. Now that M gate's going to open very quickly. Once it hits the positive 30, 40 millivolts or so, now the M gates are closed, and so now nothing can come through. Now during the repolarization phase, that's when potassium influx is occurring. And you can't have an action potential. Why? Because the M gates are closed. Now, the H gate is also closing. In fact, the same stimulus that caused the M gate to open at negative 55 millivolts or so is actually the same stimulus that is causing the H gate to close, the inactivation gate. So once we hit the positive 30 millivolts, that's when we are going to get the H gate is actually going to close. All right, so this formula right here, that's when the H gate is closed, is when you, in fact, I can just put an arrow right here, all right? Once you hit the positive 30 millivolts, now the H gate is closed, the M gate is open. And now, even though the M gate's open and sodium tries to come through, it can't go through all the way. Why? Because the inactivation gate is closed. That is blocking it from coming in. Now, the only thing that's happening is what? Potassium influx is occurring. So, well, guess what? This same negative 55 millivolts, we're just using this as an average number or so, but the negative 55 millivolts is the same voltage that opens up the potassium channels as well. It's just the potassium channels are even slower. So negative 55 millivolts happens, and what's the order? Boom, the M gate opens up, sodium rushes through. That same negative 55 millivolts, the H gate starts to close, it's just slower. Same negative 55 millivolts, the potassium channel starts to open, but it's even slower than all of this. But by the time we hit positive 30 millivolts, those potassium channels are open, and so we're getting a lot of potassium efflux that is occurring, right? Potassium is now leaving the cell very quickly, and that's why we're getting this repolarization. No more sodium entering, potassium is leaving the cell, causing the repolarization. Now, as we come back down to, say, around negative 55 millivolts or so, this M gate that opened is starting to close, okay? So as the channel is starting to reset, the M gate started to close. Once we hit negative 70 millivolts, then the H gate that's the activation gate. It was closed, right? That's going to start to open. So it has to hit negative 70 millivolts. and that's when it can start to open. So this is going to account for the absolute refractory period. So we could actually draw, we could actually just look at this right here, from this place all the way to right here, okay? This is going to be the absolute refractory period, the AR abbreviation. And why does it end? Why does it go all the way over to here? Because again, that H gate is still closed. So even though the M gate might be open, it can't go through because the H gate is closed. The H gate doesn't close until you hit that negative 70 millivolts, and then it can start to open again. Once the H gate starts to open, then if you have a stimulus, that's the depolarization stimulus is strong enough, it can pop open that M gate and it can go through. And because the H gates are starting to, are opening, and so a lot of the sodium channels are being recovered. But it's going to be harder when you're down here, you know, close to negative 90 millivolts. Well, you're further away from threshold, so you have to have a much stronger stimulus in order to depolarize the cell all the way back to negative 5 millivolts. You have to have a lot of summation in order to depolarize it even further to negative 55 millivolts. But you could do it. And so what we would call this period here, back towards resting membrane potential, this we could call the relative refractory period. I'm just using these abbreviations just for writing for you guys. They're not standard. So that's a relative refractory period. And again, you could, that's why it's relative, because you could still do it if the stimulus is strong enough. And then finally, once it hits around negative 70 millivolts, those potassium channels, that's the stimulus for them to start to close. So that's when it starts to close, and that's why we get the hyperpolarizing effect, because the channels are still open. So the potassium's still coming through, hyperpolarizing the cell. By the time it hits negative 90 millivolts, well, guess what? That's already the equilibrium, the Nernst equilibrium potential of potassium. Well, now those gates are really closed. The H gate of the sodium channels are all the way open. And now it's going to go back to resting potential. Well, why does that happen? Because of the sodium leak channels. So sodium is still coming into the cell and is bringing us back to the resting membrane potential. And now the cycle is ready to repeat itself over and over again. And this is all getting reset because we have sodium-potassium ATPase pumps on this entire cell, helping that concentration to stay the same. So this is how the absolute and relative refractory periods work, and how the gates are opening and closing and so on. Okay, and I will just mention, with all of these channels, you have other channels as well, like you have like sodium calcium exchangers and different things, but these are just the main ones. We have like mechanical gated channels and stretch activated channels and so on. But these are just the main ones that we're really focusing on. So now I want to shift our focus back over here, where we're releasing the acetylcholine onto the neuromuscular junction, and go through this a little bit. So we did a whole action potential propagation, and now we finally hit this voltage-gated calcium channel. The calcium is released, so we can release that acetylcholine. Let's just go through that mechanism a little bit more to understand what's really going on. Now, a lot of these vesicles are actually already sort of docked onto the plasma membrane right here. Okay, so actually, let's see. Maybe I want to be able to illustrate this. Maybe I'll just draw this vesicle here, and I'm going to draw some of these proteins here. Okay, this protein, we'll draw this protein, and I want to talk about what's happening. This protein here is going to be something like a synaptotagmin. Synaptotagmin, and this is actually your calcium sensor. Okay, so your calcium is going to bind onto the synaptotagmin, and that's going to initiate this entire trans-snare complex that we need to do. This is the D snares and T snares that are going to interact in order to cause the vesicle fusion to induce the exocytosis of acetylcholine. So when it binds to the synaptotagmin, there's a lot of negative charges there, and calcium sort of neutralizes those negative charges, allowing a higher affinity of this protein to the phospholipids of the surrounding proteins and membrane So it has this higher affinity, and it's going to be drawn to that. So none of this causes energy. It's all about enthalpy and the change of energy. So it's very cool. It's not even using ATP for this to occur. So when this happens, synaptotagmin does that, initiates part of this initiation. And then you have other proteins here, such as synaptobrevin. Okay, synaptobrevin. This is also a transmembrane protein on the vesicle. And then you have this protein here is a transmembrane protein such as syntaxin. And this one is a T snare. It goes on the target membrane. And specifically, it's a type of Q snare. And synaptotaxin and synaptobrevin are going to be interacting. And then we have this other protein. We'll just kind of write it over here. This is a peripheral protein. This is not a good drawing, but this one is a SNAP25. SNAP25. And this helps with this entire interaction because you have to get a 4-helix trans-snare complex that's created. And synaptobrevin is going to contribute one of those alpha helixes, and syntaxin will contribute 2 of them. And syntaxin will contribute 1 of them, and SNAP25 will contribute 2 of them. So 1, 1, and those 2 so make 4. Now you get that trans-snare complex that's being created, and it's going to fuse those vesicles and everything, all these vesicles together. And so when the docking protein, when this whole vesicle is docked, maybe it'll look something more like this. We can kind of erase this here. And then we have all these molecules of acetylcholine, right? And now the acetylcholine can diffuse into the synaptic cleft, the synaptic junction, where it can bind to our nicotinic receptor of the muscle cell itself. Okay, so that's the mechanism of release right here. Getting ACH, the acetylcholine, to bind to the nicotinic come out and bind to the nicotinic receptor. Now we talked about the repolarization of the cell membrane. As soon as that repolarization happens, then the calcium stops entering in, and so this stops happening here. Well, you still have the acetylcholine that's outside of the vesicle. Well, that acetylcholine, it gets degraded into just the 2 parts of choline and acetate, all right? And it does that by acetylcholinesterase. And the acetate just basically diffuses out into surrounding tissues and gets consumed because it's an energy source, whereas the choline will actually get uptaken back into, the choline will come back into the synaptic bulb, and with acetylcholine transferase, it'll combine with another acetate to make acetylcholine again, and then get pumped back into one of these vesicles through a proton transporter, and now you can start this entire process all over again. Okay, now the other neat thing is all of these were made actually over here, so they all had to be transported through the axon, all the way down to the end of the axon bulb. Okay. Okay, so now let's talk about once acetylcholine gets to the neuromuscular junction. So we can draw the neuromuscular junction here, and we can just kind of go like this. Say it keeps going. Here we have our T tubule. Okay, come back out. There's that imagination. Of course, that can keep going. Now we have our endoplasmic reticulum. Okay, which we can say sort of looks like this. Okay. And this is going to store a lot of our calcium, right? So we have, this is our SR, endoplasmic reticulum, sarcoplasmic reticulum, because it is a muscle cell, so it's specialized, right? Here, again, this is our T tubule. And this here is the plasmalemma, all right? Or is a plasma membrane. All right, and this, now let's go back over here, and we can write we have our ion channel right here to get sodium influx to occur. And remember that ACH is going to come and it's going to bind to this channel. This is a nicotinic acetylcholine receptor, so it's a ligand-gated receptor, and it'll allow cations to flow. So sodium is going to want to come in, but actually there's some potassium ions that will come out. And of course, you're like, wait, that's counterintuitive because that's going to repolarize the muscle cell. We don't want that. But the concentration gradients and how fast this happens in this channel, it's going to allow the influx of sodium a lot more, and it's going to cause a depolarization, making the effect of potassium not near as big. But this is like this is kind of a non-selective ion channel, cation channel, because it's much faster. And once you get the action potential to happen, you want to contract your muscles as soon as possible. And so this sort of helps with that area. So acetylcholine binds to the receptor. Sodium is going to come in, and it's going to start carrying that depolarization wave. We could have drawn it over here. It's going to carry that depolarization wave again through all your voltage-gated sodium channels until what happens, right? Until you get to, we'll draw this channel right here. Okay, not really a channel, but this is our DHPR, our dihydropyridine channel. Or it's basically it's a calcium channel. It's an L-type calcium channel, but the calcium doesn't really flow through here. But this is mechanically gated to this receptor right here. There's basically they're basically connected, and it's going to pull on this. This is the RYR, IR1, the ryanodine receptors. Okay, and this is the SR, as I said. So it's going to cause the. See, if I draw it this way, then this can happen here, and we can get calcium to release into the sarcoplasm, into the cell where you have your muscle fibers, right? Your sarcomeres, right? Your actin and myosin and everything going on. Okay. And the calcium is going to come in here. It's going to bind to troponin C, where it can start the muscle contraction. So again, voltage comes through here, gets carried through, gets sensed by the L-type calcium channel, the dihydropyridine channel. That's going to pull on the ryanodine receptor. The ryanodine receptor is going to allow calcium to enter into the sarcoplasm, where it's going to bind to troponin C and start muscle contraction. Now, one other interesting point with this dihydropyridine receptor is the cardiac muscle also has these, but it actually does allow calcium to come in, whereas skeletal muscle doesn't seem to really do that. And so in cardiac muscle, that's the calcium-induced calcium release. Calcium comes in and opens up this, whereas this is like more mechanically gated onto the ryanodine receptor. Now, once we get calcium into the muscle cell, what's going to happen? Let's walk through the whole cross-bridge cycling of the muscle fibers themselves, all right? So calcium comes in. What does it bind to? It's going to bind to troponin C. And remember, troponin is this complex that sits on the thin filament. You have your the nebulin, right? But your main filament is your actin. You have your two actin molecules, and you have tropomyosin, and then you have your troponin that sits on top of that. And the troponin has three different binding sites. You have troponin I, the inhibitory site, which binds to your actin filament. And then you have troponin C binding calcium and troponin T binding tropomyosin. Remember, your tropomyosin covers like 7 of the actin filaments, and it prevents the myosin heads from binding onto that actin. And so, yeah, this is all we're talking about right here. Now, this is an important part of this cross-bridge cycling. When tropomyosin is covering the actin, it prevents the myosin heads from binding onto the actin. Therefore, you have no contraction. You can't do anything, no cross-bridge formation. But once calcium binds to troponin C, that causes a conformational change, and it moves troponin I out of the way. So that's the inhibitory site on the actin. And it moves tropomyosin, it pulls tropomyosin out of that cleft so that the myosin can now bind to that actin. So now let's go through the entire cycle to see what's going to happen. First, you have, let's say that in this case you have a myosin is already bound to the actin, right? So this is the myosin head right here, and then this is the actin, so it's already bound tightly like this. Now, ATP that you produce from the mitochondria, or let's say you have from the M line over here, remember the M line, you have your creatine kinase, and that's going to take and basically buffer your you're failing ATP, right? You take ADP, add an organic phosphate to make ATP from creatine phosphate. So you get ATP from different ways. Okay, the ATP is going to come, and it's going to bind onto the myosin head, to the S1 portion of the myosin head. Well, not the S1, but it's going to bind to the myosin head where myosin will be able to bind to this to do ATP hydrolysis. So ATP binds, and when it does bind, it causes a conformational shift, and it allows the myosin to release from the actin. Now, the ATPase of the myosin head will hydrolyze ATP to ADP and an organic phosphate. When it does that, it causes another conformational shift, and it's going to cock it, okay? So that's the hydrolysis of the ATP. Now, it's going to bind onto that actin, but it does so relatively weak at this point. It's not strong binding. The inorganic phosphate is going to leave. When the inorganic phosphate leaves, that causes another conformational conformational change in the myosin head, and it's going to bind tight. That's the S1 configuration. So it's a tight binding, and that tight binding with basically the release of the inorganic phosphate, or that happens maybe just right after, is going to cause the power stroke, bringing that actin. So the Z disc are over here, or the Z line, right? And the actin filaments is going to pull all of those towards the M lines, basically pulling the Z disc together, right? So myosin head binds and does the hydrolysis. Then the inorganic phosphates are ready to leave, and then it pulls those Z lines closer to each other, and that causes the shortening of the sarcomere. Okay. Now it's closed, and now that the myosin head is tightly bound to that actin. During that process, the ADP has left, and it's just tightly bound, and it's going to be stuck there. It's in the rigor state is what we call it. In fact, that is actually where the term rigor mortis comes from. Like when an animal dies, then for that time they're like really rigor, right? The rigor mortis. That's because the actin and myosin are tightly bound to each other, and so it gives that rigor state. It takes ATP to release that. So ATP comes and binds to the myosin, and it releases that, and then the cycle can continue. So every time that happens, it just moves like a few actin monomers away, pulling the Z disc closer and closer and closer together, right? And then of course you have your length tension curve, so you can get more actin binding sites, and so on. So this is how the muscles are going to contract. So again, thinking about this is the calcium that's going to initiate everything. It has to first bind to troponin C in order to get the muscles, the cross-bridge cycling to start happening. At rest, when you're not exercising, you have plenty of ATP in your muscle cells, and so the ATP has bound to all the myosin heads, and then the myosin heads have hydrolyzed the ATP. It's just sitting there with the ADP and the inorganic phosphate. It can't bind to the actin because the tropomyosin is in the way. As soon as that calcium binds to troponin C, it moves the tropomyosin out of the way, and then boom, you're able to get the myosin heads to bind to the actin, and then you have we call the power stroke, right? The inorganic phosphate moves. As that causes a conformational shift, it binds tighter, and then it does that power stroke. So this is the cycle. You really want to understand that cycle, the entire cycle we went through, and remember, it's not the ATP hydrolysis that's causing the power stroke. Now we need to relax the muscle, right? We need to go back to rest. We've already talked about how the resting comes from the ATP, but we've got to get the calcium out, right? Every time, as long as that calcium is there, that calcium is just following its diffusion gradient, and it's going to just bind to troponin C. So we've got to get that calcium out. Well, in this SR, we have these pumps, right? We have these pumps called the Circa pumps. And these are the sarcoplasmic reticulum calcium pumps. And it's going to pump our calcium in and exchange it for hydrogen ions, basically. And this calcium then is bound to this protein, calsequestrin, and this binds a whole bunch of that calcium. In fact, like over 90% of the calcium is bound to calsequestrin protein, which is really important because this circuit pump, which is pumping in calcium and it's an antiport, pumping in calcium and 2 hydrogen ions going out. It does use ATP, so it's using a lot of ATP. In fact, 30% of the ATP is used by this pump alone, and it's always on all the time. So it's not like, we're trying to turn the muscle off, so we might as well start getting the circuit pump to start activated. No, it's always on. It's just that when the calcium release happens, it rushes out very quickly and binds to troponin C. So this pump is always going. Now, there's another interesting thing. You have this other, basically this other protein right here, somewhere in there. But this is called this protein is mainly in your type 2 muscle fibers, and it's this parvalbumin. And this also basically binds calcium as well. And it allows for your muscles to relax a lot faster, because that's very important to develop a fast force production and have a fast relaxation time. So parvalbumin can actually grab the calcium quickly off the troponin C and help the relaxation time go a lot faster. But most of that calcium is going to be pumped back into the sarcoplasmic reticulum through the circuit pump. And that's, again, that's how you're really going to reset the rest membrane potential. So if you go back through here, we repolarize the alpha motor neuron. You get rid of the acetylcholine by acetylcholinesterase, and now you've got to the muscle cell gets repolarized. The repolarization of the muscle cell. So of course, it's the exact same idea where you have your potassium channels. Okay, so here potassium is going to come out of the muscle cell, and that's going to lead to the repolarization. Now, the resting membrane potential of the muscle cell is around negative 90 millivolts. So you don't really have this hyperpolarization effect, which is nice. That way, when it's time to move your muscle, boom, when you get that signal, you can move it, right? So it's not like this absolute relative refractory period near like it is with a neuron. So the potassium efflux is what's causing the main repolarization. Now, it's a little bit interesting because see this T-tubule, this can get jam-packed full of potassium, and it makes it a little bit harder, and it could be somewhat problematic. And that's also why chloride ends up becoming quite important for your skeletal muscle repolarization, because chloride sort of acts as a buffer for the repolarization part of this. And then, of course, there's other repolarizing things like calcium. There's calcium sodium exchangers around here as well, helping to get rid of the calcium and to help them repolarize the entire membrane potential. Okay, so there we have it. We walked through from the beginning what the resting membrane potential, what causes and creates the resting membrane potential with the sodium, potassium, ATPase, all the different channels. Then when you get an active potential, what causes that with the EPSPs and the IPSPs, the summation, temporal and spatial summation. And then we have the myelin sheath, the nodes of Ranvier, right? The saltatory conduction that's happening, down to the axon bulb where you have the influx of calcium and then the release of acetylcholine because of this whole synaptotagmin and trans snare complex that we created to get the excitatory of acetylcholine that binds to the nicotinic acetylcholine receptor, right, ligand-gated cation channel, carrying that depolarization wave across down to the T-tubule, and then activating the voltage-gated L-type calcium channels, the hydropyridine receptor channels, and then that's going to open up the ryanodine receptors, releasing calcium into the sarcoplasm. That's going to bind onto the troponin C. That's going to move tropomyosin out of the way. Off actin, myosin can now bind tightly. It's already had the hydrolysis of ATP, so it's just ready to just start cranking away. That cycle is going to continue until we stop getting the signal over here, right? We broke that acetylcholine down from acetylcholinesterase. The neuron's already depolarized. Now we can get rid of that calcium. When we have parvalbumin, which is going to help do a quick relaxation, but these circuit pumps are working very quickly all the time. We suck that calcium back in, hold and lot it with calsequestrin, over 90% of it, and waiting now to do another active potential because the rest of the membrane potential was reset here by potassium. efflux, and of course with the use of chloride influx, so that we can reset that resting membrane potential. So you want to make sure you follow each one of these, break them down, and get those mechanisms, and you'll be good to go.